Introduce our next speaker, Dr. David Mooney, who again is a really eminent figure in the field. Just very quickly, David is the Pincus Professor of Bioengineering at Harvard in the School of Engineering and Applied Science. David also is a founding and core member of the VIS Institute, and also he is really a leader in biomaterial engineering for tissue regeneration and also immunotherapy. And last, I think in 2019, he was voted by the Nature Biotech as a top 10 leading translational bioengineer. With that, we welcome Dave to tell us about his newest findings.
So, well, thank you for the introduction and for the opportunity to come and participate in what's been a really exciting program, and I'm looking forward to learning a lot over the next few days. I'm going to talk today about one facet of the work in the lab today relating to adoptive T-cell therapies. Before I get started, I should mention that the work I'll describe today has all been funded by the NCI through a couple of different activities. There is a commercial engagement in the first part of my talk where that led to the founding of a company, Immulis, that was then purchased by Lyell last year, and so I do have a commercial engagement with some of the work that I'll describe in the early stage of my presentation today. Now, as I said, I'm going to talk about T-cell therapies, and as most people here in the audience are probably familiar, there's been tremendous excitement about adoptive T-cell therapies. The idea that you would take T-cells from a patient, typically genetically modify them, in this context on this slide, to make a CAR T-cell. So it's capable of recognizing and destroying cancerous cells that have that signature that you're training them to recognize. And this is actually an image from one of the first patients that were treated now a number of years ago with CAR T therapy. And you can appreciate how a single infusion of these cells led to clearance of the cancer throughout this patient. It's been really spectacular results, has led to several FDA-approved CAR T therapies. And there's been an explosion in the number of clinical trials and products being developed in this general T-cell space, as shown here in this graph, which just shows basically T-cell therapies in development up until a couple of years ago. So this is a very active space. And on the one hand, there's been tremendous success, but on the other hand, there still are significant challenges. And so I'm here today to talk to you about some of those challenges that exist with this approach to therapy. Now, to help you kind of understand what we're trying to do, let's go back to how these therapies work. So as I mentioned a moment ago, the approach is that the cells are isolated typically from the same patient, although those allo products that are under development. These cells are then activated outside the body, genetically modified if it's basically going to be something like a CAR product, and then the cells have to be expanded. We need large numbers of cells to treat patients. And then these patients are reinfused back into the individual with cancer. So the ex vivo manufacture of T cells requires T cell activation and expansion. Now, the T cell expansion part of this can take weeks to months to generate sufficient numbers of cells to treat a patient. And a certain fraction of patients will actually die before sufficient cells are generated to treat them. So this creates, obviously, an impediment to the use of this therapy. In addition, this long manufacturing process is part of the reason we have such a high cost associated with these types of therapies today. Now, beyond that, once the cells are infused, while overall, in certain types of blood cancers, they work well, there is a significant variability in that you'll have, as indicated in the slide, you'll have some patients that will be complete responders. You'll see expansion of these T cells after they're infused into the patient. They'll be very effective at clearing the cancer. At other times, you'll have partial or non-responders as well. And overall, these therapies, while they've been successful in blood cancers, have not been successful to date in solid tumors, which obviously is a big component of the total cancer burden here. So some unmet clinical needs then are to improve the quality and yield of T-cell products. So basically, can we manufacture these cells better than they're manufactured currently for clinical use? And then, really importantly, and probably more importantly, can we enhance their therapeutic activity after infusion? So I'm going to talk today about two stories. Most of the time, I'll be describing efforts that were underway to try to improve the manufacturing of T cells and get the right type of T cell at the end of the manufacturing. And then the last part of the talk, I'll give a couple of short stories about how we're actually trying to actually then alter the behavior of the cells once they're in the body and further improve their effectiveness. So that's how we'll organize things today. Now, to take a step back, for those of you who are not familiar with T cells and T cell activation and expansion, how T cells in the body are typically activated and induced to expand in numbers is they engage with an antigen-presenting cell, as shown schematically on this slide. So the antigen-presenting cell on the bottom in purple will directly stimulate the T cell receptor and also provide co-stimulation. So it's typically called signal 1 and signal 2. The antigen-presenting cell will also secrete various agents that act in a paracrine manner on the T cells to control their proliferation and their function. Now, one can manufacture T cells by isolating antigen-presenting cells from a patient in addition to the T cells, but now you're basically dealing with two types of cells that you have to put together. It makes the whole process much more cumbersome, and oftentimes, if you're taking cells from a patient, the APCs may not be very functional. So instead, what's done commercially is to use an artificial antigen-presenting cell. And the industry standard today is a particle called Dynabeads. So it's a solid polymer particle, and to provide the right stimulation of the T cells, antibodies that can bind to the T cell receptor and provide the co-stimulation are covalently conjugated to the particle, and then these particles are added to the cell as a mimic of a normal APC. In addition then, one feature here then is now instead of presenting these engagers of the T cell receptors from a cell in basically a cell membrane, where a key part of the process is the T cell actually engages with these, physically remodels, and some of you may be familiar with, creates a synapse that's believed to be very important in the activation process. Here that's not going to be so feasible because these ligands are presented in a mobilized manner on the particle. In addition, for the paracrine acting factors, what's done with Dynabeads and other commercial products is these agents are simply added to the cell culture media. So they're not being signaling, they're not being engaged with the T cells in the normal manner. So we became interested then of whether we could develop a new type of artificial antigen presenting cell a couple of years ago that would more closely mimic key features of the APC T cell interaction. And that system is shown here schematically, and this is work that was actually started by Alex Chung, who's now actually heading up the efforts at Lyle to commercialize these products, and then David Zhang, PhD student in the laboratory. And the basic approach is we start with a microparticle instead of a micron-sized cell. We specifically are starting with mesoporous silica, and these mesoporous silica particles look like this. Typically aspect ratios, or I should say lengths of 50 to 100 microns, widths of about 5 microns. One of the attractive features of these particles is that they have long aligned nanopores that you can appreciate in the cartoon. So this creates a convenient place to park biological cargoes for their sustained release to mimic the paracrine-like engagement of these particles with cells. And here in the bottom, just showing release kinetics of a few of the different types of agents that one might typically want to engage with T cells. So we can have a sustained release for multiple days from these particles. Now to mimic something that then physically is more like a cell, we coat these with liposomes. We create a supported lipid bilayer on these particles. And what you can appreciate from this video is a FRAP study where we basically photo bleach the fluorescently labeled lipids on the surface of the microparticle, and you can see we get this recovery showing you that it's a fluid-like system on the surface of these particles. So similar in some ways to the surface of a cell. Now, unlike a normal cell, where it's going to have a certain amount of stimulatory cues, basically things that can engage with the T cell receptor and provide co-stimulation, here with this artificial cell, we can precisely tune in the amount of stimulation we want to give from each particle to the engaging cells. And so here's just some data showing you how we can basically tune in the quantity of the stimulation from the particle. And then finally, once we have our particles, the idea is we take these microparticles, we combine them with T cells. Here the T cells are in red. The T cells can then engage with the cues presented from these microparticles, and we hope get appropriately activated and proliferate. So that's the system that I'll be using for the first about half hour of the talk today. Now, a key feature of this system is that we can do both basically a polyclonal expansion of T cells, so kind of expand any T cell that's present in the system by presenting antibodies that specifically bind to CD3 and CD28, and that system is shown on your left. But we can also do antigen-specific expansion by instead conjugating MHC with the right peptide antigen to the system. And I'll give you some demonstrations of both, and clinically these are used in different contexts, so I won't go too much into that today. Okay, so does the system work in terms of being able to expand T cells? So this is data from one of the very early experiments. So we took these microparticles, we combined them with T cells, and what you can appreciate is over time, the T cells physically engage with the particles, they adhere to them, they move them around, they cluster them, and they form balls of T cells around these microparticles, as you can see from the image here. If we look at the proliferation of the cells, what we find is that we get substantial expansion of the T cells when they engage with these microparticles. Also shown in this graph is dynabead expansion for comparison, and you can appreciate that we get a substantial increase in the expansion rate of the cells with these particles as compared to dynabeads, so we can basically manufacture the cells more rapidly with the system. Now, if we manufactured the cells more rapidly, but they were dysfunctional, we typically talk about exhaustion of T cells. When they become dysfunctional, then we wouldn't really be doing anything useful. So we'll get more into this actually later in the talk. But as a first early metric, we analyze the cells for expression of a few cell surface markers that are known to be representative of T cells that are becoming exhausted. And what you can appreciate is that even though we're proliferating the cells quite a bit more with what we're calling this APCMS system, that if we look at markers of exhaustion, PD1 and LAG3, that we actually very similar to dynabead expanded cells. So we can grow the cells more rapidly without actually apparently harming them. Now, a key question when we first got started is what element of the system is actually beneficial? Is it the fact that we have more of a cell-like interface, that we're presenting these cues and the cells can mechanically reorganize the ligands? Or is it the paracrine-like activity in terms of release of these agents? So we did a really simple experiment that's shown here, where we compared expansion with dynabeads to expansion with these microparticles that did not include any of the cytokine. So we just added the cytokine to the media like one does with dynabeads. And you can see in this context, we got a significant enhancement in proliferation. So the more natural presentation of the ligands is important here. But then what you can see in the last condition is if we compare to the APCMS that does have the IL-2 released from the microparticles locally, we get a further benefit. So both elements seem to be really important in the activity of these microparticles and their ability to promote T cell expansion. Now that was in the context of polyclonal expansion. As I mentioned, we can also do antigen-specific expansion of cells. So in this case, we take MHC, we take the right peptide, we present it from the MHC on the surface of these particles. Here I'm going to show you some data where we were specifically looking at expansion of T cells that would be specific for Epstein-Barr virus. And we chose two peptides that are known to be efficient antigens for generation of T cells in patients. So then we collected basically T cells from patients. We screened for HLA-A2 in prior EBV exposure, indicating that the T cells should be there in some numbers. And then we'd isolate, and then we'd culture with these particles. And then we would look to see if we could actually expand and enrich these specific T cells of interest. And so that's some of the data that's shown here. And what we're looking at is expansion on the left of the T cells that would be specific for CLG. And we basically have APCMS with CLG. And you can see we get a really robust expansion. We get about a 4,000-fold expansion of the T cells. Now, what's really striking here is if we compare to the normal way that these T cells get expanded in the body, antigen-presenting cells, which we also isolate and use in the system, you can see that the the monocyte-derived DCs presenting the same peptide lead to about a 20-fold expansion. And then on the right, you're just looking at expansion of the GLC-specific, and in both these we have the other peptide as a control. But what this data shows you is we can do antigen-specific expansion, and it dramatically outperforms the normal antigen-presenting cell that we isolate from the body. So it looks like the system is actually quite potent in being able to expand the T cells. Now, of course, we're very interested in the function of these cells, and so early on we then went ahead and basically did the key test, which is to go back to the standard preclinical models that were used to develop these CAR T therapies originally. So we use immunocompromised animals, we introduce human cancer cells, and then we expand CAR T cells that are specific for this type of blood cancer. And we do it either with the APCMS or the dynabeads. We deliver the same number of cells, of T cells, and see if they're equally effective. And what you can appreciate from the normalized total flux, which is indicative of the amount of cancer cells in the animal, or the survival, that the T cells from both expansions are effective, and actually the T cells from the APCMS actually are statistically a little bit better. So we can now manufacture cells much more rapidly, and it looked like, based on this data, that actually we could get a little bit more function out of the cells as well. So this was the first part of what we explored. However, there's a lot of variable space here that we can play with to try to further improve and control the T cells. And I'm going to give you a few examples of that. So one is the microparticles themselves. So a cell, basically we isolate from the body, we take it as it is. These microparticles, we can do something as simple as changing its size. We can control the size of the pores, these nanopores that run through. We can also control how rapidly they degrade. And these will, I didn't mention earlier, but will simply dissolve over time. So at the end of the expansion and manufacturing, we don't have to remove the dynabeads as is done commercially. Here the particles are just gone. So there's no separation process at the end. But in any case, the way these are manufactured is basically surfactant templating. It's all aqueous chemistry. We add a surfactant, self-assembles. We then condense silica around the particles, and then we remove the surfactant to create these particles. So there's a number of really simple variables that one could manipulate in this manufacturing process to control the physical properties and some of then these features of the particles. So we took a very generic approach, non-biased approach, and simply varied these different elements in the manufacturing process, created a whole family of different types of microparticles with varying physical properties, and a few of these are shown here. And then we simply screened to see how effective they were. So for example, we screened to see how effective they were at maintaining the lipid coating. And you can see that each one of these individual data points is a different manufactured particle. And you can see they varied quite a bit in how effective they were in terms of the lipid coating. They varied tremendously in terms of their ability to provide a sustained release of various cytokines, such as the IL-2 here. And then they also were varying quite significantly in their ability to then expand T cells. And actually for reference, the reference that's pointed out here in the T cell expansion is the initial particle for the data I just showed you. But you can see with this very empirical approach, we're able to now get about a 50% improvement basically in the expansion of the cells by having a more optimized physical particle. So that was promising and basically showed us that we could manufacture cells more quickly. What we're very interested, though, is going beyond just making more cells or making them more quickly and saying, can we actually program T cell attributes to get the best cell possible? Now, for those of you that are not in the cell therapy space, I'm going to put this in context. So if we think about therapies in very broad classes, traditionally we had small molecule drugs. These had a completely defined chemical structure, right? So we knew exactly what it was, and presumably it would, basically be very similar in each patient. As we heard about in Dr. Langer's talk, then we have things like macromolecular therapies, we have proteins, we have mRNA, we have other types of agents we use as therapies. Those have a more complex structure. In the case of proteins, for example, we know that we may have some varying glycosylation patterns and other features, but generally speaking, we know what the molecule is we want to deliver. Now, in contrast, if we start thinking about cell therapies, a cell can have many, many different states, right? Cells can be at different stages of differentiation. They can have different stages in context of T cells, exhaustion. And so the question becomes, what cell state do we want? And then how can we actually manufacture a cell to have the right state when we transplant it into a patient? And these are things that are largely unknown, though we're starting to get a variety of clues from a number of preclinical and clinical studies. So for example, here we're looking at some data on the left from a preclinical study and on the right from some clinical studies. And on the graph on the left, it also along the top is helping you to kind of demonstrate how T cell differentiation occurs, where we have different basically more differentiated T cells that have greater effector function, but less stability in the body as we go from the left to the right. And what this graph shows you is a really simple experiment where in the same cancer model, investigators took differing, or basically the similar numbers of T cells at different stages of differentiation, either with one population or mixtures of populations, and then introduced them into this model I just described to you a moment ago. And what they found was vastly different outcomes. So the differentiation status of the cells and the ratio of CD4 to CD8 cells made a huge impact in terms of how therapeutically effective the cells were. Now, the data on the right from clinical trials, basically, so these patients were analyzed after the fact, and they looked to see if the T cells that they introduced, what was their exhaustion status at the time of delivery into the patients. And what they found was that the cells that had a higher profile for exhaustion led to more partial and non-responders in following delivery into patients, while the cell populations that had low exhaustion tend to have more complete or partial responders. So the state of the cell is clearly really important, and the question becomes, well, how do we actually control that as we're basically manufacturing these cells? So we think that this system could be a really nice one to enable this control, because we have a number of features that we can regulate, right? So we can control physical properties of the MSRs we just talked about. We can vary the mitogenic cues. We can vary the lipids. We can control the T cell signals from these particles. And we've actually been exploring this whole space. I'm actually going to just talk to you about kind of the simplest possible version of this, which is basically controlling the T cell signal. I'm not even going to present data on the mitogenic cues. So we simply vary the amount of stimulation we provide, basically the quantity and the density of the antibodies. We then took human T cells from donors, and we expanded those cells, and we looked at basically their proliferation, the differentiation status, cytotoxicity, cytokine response, survival, and then looked at a number of features basically indicative of different aspects of T cell phenotype that we believe, based on previous work, are going to be really important. So took a completely non-biased approach to basically create different versions of this MSR, and then we, again, as I said, expanded T cells and monitored basically the T cell product. And so what this data represents here on the right is, for those of you familiar with analysis of gene expression data, we oftentimes try to non-dimensionalize it using principal component analysis. So we've done the same thing here. Each one of these blue dots is a different manufacturing run of T cells, where all we've done is varied basically signal 1 and signal 2, density and amount on the microparticles. And what you can appreciate from this is that we get a broad array of different types of T cell products resulting at the end of this process. Now, we can do typical types of analysis that one does in gene expression analysis. In this case, basically identify clusters of, or basically groupings of these T cell products and analyze their features. So for example, if we look at the amount of expansion in these different K clusters, or we look at the resulting CD4 to CD8 ratio, we can look at the exhaustion markers, PD1, TIM3, or we can look at the differentiation status, looking at the ratio of effectors, effector memories, central memory, cells. And what you can appreciate is we can take the same starting T cell population and we can generate vastly different T cell products at the end of this process simply by controlling features of these artificial cells that we use to expand the cells. Okay, so we can do that. I'm not going to get into a lot of the detail here today, but one of the really key features is basically the amount of stimulation is actually a key element here, is indicated by the color coding on the principal component analysis. So now all that was done with healthy cells. What about if we now move to cells actually from patients, which are the ones we'd actually want to be able to expand? And so this work was all done with Kathy Wu and Jerry Ritz at the Dana-Farber Cancer Institute. And in addition to David, also Nico, a postdoc in Kathy's Wu, was working on this project. Sid, who's a PhD student at MIT, and Kwasi, a PhD student in the laboratory, did this work. And we basically were taking cells from acute lymphoblastic leukemia or acute lymphocytic lymphoma patients, and then basically putting them through the same process to try to understand what were the differences between normal T cells from healthy patients and T cells from these cancer patients. One of the first things we find is when we initially isolate the cells they actually are quite different in terms of the CD4 to CD8 ratio. And if we look at basically the differentiation status of the T cells that we actually isolate from these patients and compare basically to T cells from healthy patients, again, we see really big difference. So the starting T cells are different. And the other thing you can appreciate from the individual symbols here are individual patients. You can appreciate that not only is there a big difference between healthy and diseased, but within the cells from diseased patients, there's tremendous variability. We get very different cells when we get them from different patients. Now, if we take the APCMS and we basically use them to expand the cells, we're comparing here basically a variety of doses. The D is dynabeads, the A is APCMS. Just for those who like to have a direct comparison, the red bar basically indicates dynabead and APCMS that are matched for the amount of stimulatory signals. So now if we actually look to expand the cells with the system to basically get enough cells to use therapeutically, we find a really significant difference in how these cells expand when they're derived from these patients. So with the dynabead expansion, the expansion is actually really poor. We actually generate relatively low numbers of cells. When we use the APCMS, we can get much more significant manufacturing of the T cells. But what's really interesting is you can appreciate that with these cells, there's actually some optimal expansion at some intermediate level of stimulation. Too little stimulation we know doesn't actually lead to anything effective, but with these cells from diseased patients, too much stimulation is also really a negative. Now, we find that not only is the expansion of the cells, but the function of the cells is actually dramatically altered as well. That when we take these T cells that we've expanded from the diseased patients, and we use them to kill basically their target cells in culture. So basically a cytolysis assay, in this case in culture, you can appreciate that as we vary the ratio of the effector to the T cells, basically we get different curves there, which indicates that basically we have very different functions as we vary the amount of stimulation of these cells. So the number of cells, their function is dramatically impacted. Now, of course, what we really care about is not how these function in vitro, but how do they actually function back in the body. So we basically picked 3 representative basically batches of these cells that we manufactured from the same patient's initial T cells. In this case, the 0.1, 0.15, and 0.3, which just indicates how much stimulation we provided. And then we went ahead and basically took these and transplanted them into that same animal model I described before. And what we find is not only is their phenotype different outside the body, but it's also dramatically different inside the body. So here, for example, if we look at the number of CAR T cells that are present in the circulation of these animals, as we go from the low to the mid to the high, and we also look at the distribution of their effector to stem-like differentiation status, we see dramatic changes depending on how we stimulated and manufactured these cells outside the body. If we look at the here's the differentiation status in terms of, we also see dramatic differences. So not only are the cells different before we put them in the body, but then they're very different after they're actually in the body. And this just basically provides a kind of a summary that basically at the low versus the medium versus what we're calling the high, we have dramatic differences in the CD4 to CD8 ratio, the differentiation status, their markers for exhaustion. And then if we take the same number of these T cells and introduce them back into the same animal model, so a humanized mouse model containing human cancer cells, we find the same number of T cells that we manufacture differently have dramatically different impact and success. So as you can appreciate, if we do a mock, basically no treatment, the animals succumb to the cancer. If we have the high level of stimulation, even though it's the same number of T cells, it's modestly effective, and actually the animals all relatively quickly succumb to disease. But if we have the lower, the medium stimulation, we get a much more benefit from the same number of cells. And you can appreciate that from the basic quantification of the total flux and the survival in the bottom. So what this is telling you then is that depending on how we manufacture these cells, we can make them either highly effective, highly ineffective, or something in between. Now, the challenge here, as I mentioned earlier, is that each time we get these cells from a patient, they're different. So how do we start with something that is different initially and then turn it into something that's the same at the end so we get the right types of function? That's actually pretty complex. So the approach we've been taking to explore this is to use AI approaches. And the general idea is that we want to characterize the T cells when we obtain them from a patient, determine what their function is or what these characteristics are that have been, some of which I've been describing, some of these cell surface markers, CD4 to CD8 ratio, things like this. And then we know what we want at the end, at least for right now. That may change over time. This has not been established clinically. But the premise is we can eventually identify exactly what attributes we want. And then we basically take a training approach where we take all the data we've been generating with different patients and we simply explore how the way we stimulate, the way we design these artificial cells, how it connects the starting T cell population to the ending T cell population. And we've been able to do this now with about 20-some patients. This actually is just the first handful of data that we generated. You can see that this actually approach, which ended up just being a simple random forest regression model that we used here, was able to actually give us a pretty high ability to predict the exact stimulation we wanted for a particular patient's T cells. You can appreciate there's some variability. Some patients we match up better than others. But overall, what we're finding from this limited number of data that we have to date is that we can indeed, based on the starting T cell product, or excuse me, based on what we want for the product and what we know is the input, as we look at different cells from different patients, we can actually predict exactly how we want to change the APCMS to give us that desired product. So this still has to be validated in hundreds of patient samples, not tens, which is where we are right now. But we think this could be a very potent approach to actually personalize these CAR T therapies to specific patients and hopefully dramatically increase their effectiveness overall. Okay, so that's one aspect of what we've been doing. Now I'd like to change gears, yeah, because I've got some time left, to talk about once we introduce these cells into the body, is that it? Is our control done? Or could we think about actually having control after the cells are in the body as well, beyond kind of their attributes when we introduce them? So we'd like to actually be able to, for example, if we have a CAR T product that's failing, could we rescue that by having some intervention after the CAR T therapy? And the approach that we've been pursuing initially, and so it'll be the first of two approaches I'll describe to you, is to actually use the same exact system, these artificial antigen presenting cells, and introduce these into the body to create a depot to stimulate the T cells that are already there, that we've already transferred. And the idea is we'll simply do a subcutaneous injection, for example. The T cells that are circulating can come to these particles, get activated again, get proliferated again if needed, and then hopefully enhance their therapeutic efficacy. So a really simple idea, and this is work being done by David and then Josh Brockman, a postdoctoral fellow in the lab. So of course, the first question is, are these particles, is the system even stable in the body? So if we take fluorescently labeled lipids, we basically create the system, we inject it, we find that actually the lipid on the system actually is relatively stable, that we basically for several weeks we can still have a fairly significant amount of the supported lipid bilayer present. This is just an image of one of these at explantation. They rapidly become vascularized, which is also key, because if there's not vascularization, there's no way for the T cells to traffic to these devices. And actually this is going to be this is a FRAP video. I don't have a way of triggering it right here, but basically this would show you that we still have a fluid-like supported lipid bilayer on these particles, even when we remove them after 21 days. Now, in terms of directly looking at the bioactivity, the approach we took initially was very simple. We simply actually removed the particles from the body at different times after introducing them, and then added these to T cells in culture to see if they could stimulate T cells. And that would tell us if they're still bioactive. And what you can appreciate here, we're looking at basically a CFSE dilution, for those of you familiar. Basically here, the lower the number on the left, the more bioactive it is. And then we looked at a specific marker for activation, expression of CD25. And what you can appreciate is the particles that we pulled out up until about 14 days were still bioactive. Beyond that, we actually didn't see any bioactivity. So it looked like we could inject these and they would still stay active for about 2 weeks. Now, of course, that doesn't ask the question whether T cells that are circulating will actually come to that site, get stimulated and leave. So we've examined that as well. So here we're looking just as a metric of that for the number of circulating CD3 cells in the animals as a function of time after we do this boost. And what you can appreciate when we have the system that has the cytokine IL-2 and both the anti-CD3 and CD28 antibody, we get a significant increase in the circulating T cell number after we do this boost, suggesting we might be able to then enhance or perhaps rescue this failing product. To test this directly, we again went back to the same model, but here we had a couple controls. We basically did nothing. We took these materials that only had the cytokine, and then we took the system that had the stimulatory antibodies and the cytokine, and in this case, we're purposely introducing a subtherapeutic CAR-T dose to kind of mimic a failing product in a patient. And what you can appreciate from the data here, if there's no treatment, the animals succumb quickly. The CAR-T cells alone, there's some benefit as we expect, but not a long-term benefit. If we have blank particles that don't have any bioactives, there's no effect. The IL-2 by itself gave a benefit, but if we have basically the stimulation, co-stimulation from the particles as well as the IL-2, we get a dramatic enhancement in the effectiveness of these same number of initially transferred T cells. So it looks like we can indeed have a significant impact on the T cells after they're introduced in the body with this relatively simple approach. So I'm going to wrap up just this part of the talk here before I talk about one more thing. And hopefully what I've been able to demonstrate is how we've been able to design a really simple artificial antigen presenting cell that we basically can have some really straightforward inputs, soluble cues, things like interleukin-2. We can have surface cues, and we can basically use this to manufacture T cells, or else we can actually do this directly from PBMCs, though I didn't show you that data. Now with this, we can actually more rapidly expand the T cells. We can actually tune the T cell product and get highly functional cells even out of suboptimal starting T cell populations. So we think that this will dramatically impact the time, the control and the cost and the overall effectiveness of these adoptive T cell therapies. That obviously has to be validated still in clinical trials, but we're moving towards that at this point in time. More broadly, though, we think that this could be a really useful platform in a variety of other settings. So there might be a variety of different inputs that we could provide to basically control cell behavior. And we think this general approach could be broadly useful for manufacturing a number of other types of cells that are interesting therapeutically. So many of you are probably familiar with NK cell work that's being pursued quite avidly in the clinic today. We've now demonstrated that we can use a variation on the system to basically do 10,000-fold expansion of NK cells in a relatively short period of time, which is addressing one of the key limitations of those cells. We're also looking at T cell progenitors and demonstrating we can use this to generate large numbers of those cells as well. So we think this could be a broadly relevant system for manufacturing a variety of cells in the future. Now, I'd like to take the last few minutes and talk about a similar aspect, but we've become very interested in kind of T cells and T cells therapies in the context of solid tumors. And as I mentioned at the start of the talk, while the current products are approved for blood cancers, they do not tend to work very well against solid tumors. We actually have a number of different approaches we're currently trying to explore to enhance the efficacy of adoptive T cell therapies in the context of solid tumors. This slide just kind of gives an example of some of the barriers that T cells face, specifically in the context of solid tumors. I only have time today to talk about one strategy that we're pursuing, but I decided to talk about it because I think it actually complements well what I've just described. So in the last part of the talk, I described how we could introduce these APCMS particles that release a cytokine. Well, what we've recently been exploring is whether we could actually maybe simplify this and actually just deliver the cytokine itself. Now, as many of you are aware, cytokine therapies have been widely explored in the context of cancer therapy. They tend to be severely limited by toxicity issues. So there's a variety of cytokines that have been identified that could be useful by themselves or as a co-therapy, and just a few of them are listed here, but they tend to lead to lethal toxicity, inflammatory signal, they're cleared very fast, so it's been a really significant limitation of their use. Now we can potentially provide these agents from a local depot, as I showed you with the last little bit of the talk, where we combine that with direct stimulation of the TCR and co-stimulation. But we also became interested in whether we could just directly provide cytokines in concert with the T cells. And our work here was actually inspired by some work that had been going on, basically Professor Chang at the University of Illinois, where he demonstrated a few years ago, along now with many others, that one could use metabolic labeling to introduce distinct and orthogonal functional groups onto the membranes of cells. And the general idea here is that one takes artificial sugars, for example, artificial sugars that have a click functional group on it. You can simply feed these to cells. They will use them in their normal metabolism, and depending on the sugar, it will get incorporated in different ways within the cell. And in particular for azido sugars, which is our focus here, they end up being utilized in the formation of the glycocalyx. So now you end up with a situation where you have a very high density of these orthogonal chemical group that we can target that's now present on the surface of the cells. Now, to kind of put this in contrast to other approaches to try to target cells that we're all familiar with probably in this audience, things like antibody targeting, here we can get much higher densities of these cues on the cells, so create many more ways of basically introducing agents to the cells. The targeting efficiency ends up being very high, and because we're talking about small molecules here, we actually don't tend to have concerns about immunogenicity from this component. So the approach we've been pursuing is a really simple one, and actually is derived from some work we did a few years ago with in vivo metabolic labeling. Here, though, we're doing it in cell culture, and the idea is we take T cells, we simply add these unnatural sugars. In this case, we've been adding them in the context of nanoparticles that we synthesize. They basically get taken up, these nanoparticles get taken up by the cells, metabolized, and the idea is we then have azide-labeled cells. Now, once we have the azide labeling, we can then simply click on cytokines to get cytokines directly conjugated at whatever density we would like, up to some maximum ceiling, onto the cells. Now, the reason we think this is really appealing approach is that this is really simple, that this would fit right into current T cell manufacturing processes. We wouldn't have to make something else and combine it with the T cells. We literally just add these unnatural sugars to the cell culture media, and at the end, we have the cells that are labeled, and then we basically can click on whatever we want in the cell surface and wash away what's unbound. So it would be minimally impacting the T cell manufacturing processes that I've described to you to this point in time. This is just an image of basically seeing the azide functionality that we've visualized here around an individual cell. You might be wondering about the stability of this. So we've looked at the stability of the labeling. So we've labeled the cells, washed away the unnatural sugars, and what you can appreciate is you get significant amount of these sugars present for about a week. So we have about a week to manipulate the cells with this really simple approach. And so we've been now starting to explore how does this impact T cell behavior. So we've looked first of all in cell culture. And these graphs are a little bit complex. I'm sorry these are not as polished as the other ones I presented, but I'll kind of walk you through it. We've looked at basically a few different things. We've looked at T cells that basically we labeled but didn't add the cytokine. We've looked at T cells that basically have IL-2 just added at all times in the cell culture medium. It's just continuous IL-2 supplementation. We have basically T cells that we added the cytokine just for the same amount of time that we do the labeling and then washed it away. And then we have basically T cells where we conjugate the cytokine and then wash it away, and then we look at the function of the cells. And if we look at a variety of metrics of the T cells, for example, their CD4 to CD8 ratio, their expression of CD69, PD1, what you can appreciate is that the transient exposure doesn't do much to the cells. The continuous exposure changes the T cell phenotype as we would like. But what's really interesting, if we conjugate the cytokine and then wash away, we actually have very similar effects to continuous T cell supplementation in the media. And actually, if we look at the function of the T cells in terms of their ability to kill the target cell, it actually is pretty much identical between just having the cytokine continuously present versus labeling it on the cell surface. So to see whether or not this would actually have any impact on the T cells in vivo, we went to a solid tumor model, the B16F10 that many of you are familiar with. We did adaptive transfer of basically P-mel T cells that we conjugated IL-12, and we kind of looked to see what happened. Well, one of the striking initial features we found was this really simple approach dramatically altered the ability of the T cells to actually enter the tumor microenvironment. So here we're looking at the number of T cells that were transferred in the tumor microenvironment, and you can appreciate the T cells by themselves, they don't really get in, but basically if we have this IL-12 conjugation, we now significantly increase the number in the tumor microenvironment. If we look at the function of these T cells, by looking at their expression of a variety of cytokines that we know are associated with basically killing function, and so we're looking at, on the right, some of these different cytokines, and then it's color-coded where red means they express more, blue means less. You can appreciate when we have the T cells with the conjugated IL-12, we dramatically increase many of the key cytokines. The comparisons here is we also just take the same amount of cytokine and deliver it in soluble form with the T cells to kind of mimic typical co-therapy, and then we basically have the transient again in the T cells by themselves. Now, so we basically change how the T cells that are transferred function. We also change how they engage with the host immune system. So for example, if we look at the antigen-presenting cells and the draining lymph nodes from that tumor, we see a significant increase in the expression of markers indicating those normal APCs are activated. We see increased MHC2, CD86, in both the CDC1s, which are important in cross-presentation, some of you may be familiar with, to generate cytotoxic T lymphocyte responses, as well as the CDC2s. So we dramatically alter how the host immune system is engaging with the tumor with this really simple manipulation. Further, if we look at whether or not this results in a host immune response to the tumor, we then did a similar study, but here we actually use B16F10 that express an artificial protein, OVA. So the T cells we transfer don't recognize OVA, but we look to see whether we generated host T cells that basically recognize OVA. And what you can appreciate, when we have the conjugated IL-12 on the T cells, we get a significant increase in the host T cell specific against the tumor antigen. So now we're not only having the T cells we transplant be more effective, they're marshaling a host immune response against the antigens in this tumor as well. The net result of this is that we can have a significant impact in terms of the success of the T cells in terms of fighting cancer. Here we're just looking at basically delivery of a small dose of cells, and you can see that when we have the conjugated IL-12, it makes the T cells much more effective. And we also, as shown in the bottom right, are generating basically a significantly greater expansion of the T cells after transplantation into the body with this simple conjugation. So I'm going to stop here. And hopefully what I've been able to demonstrate is how we're trying to design really simple strategies to enhance adoptive T cell therapy. Now, I do want to put this in the context of some broader work in the laboratory, so you can kind of see how this fits into a bigger approach to T cell biology in the context of cancer. And we're very interested in manipulating T cell biology basically from the beginning to the end. So, though I didn't have time to talk about it today, T cells basically originate in the bone marrow, where hematopoietic stem cells will differentiate into common lymphoid progenitors. We have some biomaterial strategies now. Oh, wow, actually, I just realized, okay, this slide changed dramatically in the transition to the computer, so they had me go through this before I gave the talk, and I didn't notice that this slide was changed. So the images here are largely wrong. But anyways, what this would show you is that we have biomaterial strategies that enable us to enhance T cell reconstitution by specifically enhancing differentiation of hematopoietic stem cells down the common lymphoid lineage. We're developing engineered thymic tissues, and the thymus is where T cell progenitors go to differentiate into single positive CD4 and CD8s. So we're developing engineered thymic tissues. We're also developing vaccine approaches. So we heard a wonderful talk on vaccines from Dr. Langer at the start of the meeting. We're using biomaterial strategies to create therapeutic vaccines against cancer. And then the last part of this is what I've just described to you, is how we're developing material-based technologies to try to manipulate adoptive T cell therapies as well. So I'll finish up there, and I'd be happy to take any questions.
Thank you. I'm Ángeles Vaquerizo. I'm a transplant surgeon, and congratulations for an amazing work and presentation. I have a question regarding the exhaustions of the T cells and the markers. I see that you check the PD-1. Did you check the PD-1 ligand?
So on the in the T cells, no. We're looking for we look for PD-1 in the T cells. Yeah.
Because let me tell you, I have done a study in resected liver cancer.
Yep.
So the PD-L1, most of the time, you just analyze in the tumor cells. I found only 11% presentation of PD-L1 in the tumor cells, and like 50%, 45 to 50% in the tumor-infiltrating lymphocytes. And that is what I was thinking, that probably that is the marker of T-cell exhaustions. Anyway, it's just a thought.
Yep. Yeah, so you're raising a really important point here, which is that, so what I was using today, I was using expression of specific cell surface antigens as a marker for exhaustion. But that's actually not exhaustion. These are markers. And we use the ones that are fairly standard. At the end of the day, for you to really say that we have a T cell in an exhausted state, it really is a functional measure. So some of the functional measures we have are consistent with that, but yeah, the marker is only an indication. It's not an absolute indication of exhaustion. Yep.
Great talk. I have a question regarding the mechanical property of this artificial APC you discussed. So the native APC, the mechanical property is about a kilopascal. Have you manipulated the artificial APC in the mechanical property and see whether that affect all of these T cell functions?
Yeah, so we've not manipulated it here in terms of the mesoporous silica particle. What we have done is the T cells are engaging with the receptors that are presented from the supported lipid bilayer. And so we've done a pretty significant amount of studies where we've altered basically the bilayer composition and looked at lipids that have a very wide range of melting temperatures. That will end up yielding a more solid-like versus a very liquid-like interface for the cells as they engage with these receptors. And there is a dramatic impact on the physical properties of that supported lipid bilayer on the resultant T cell basically activation and expansion. And not surprisingly, there is an optimal, which is actually the lipid composition we're using here. Basically, if there's too little resistance to the T cells when they engage with these receptors, they don't tend to respond. On the other hand, if we make it so solid-like that they can't easily remodel the ligands and make a synapse, then it's also not very effective. So that's what we've done until this point in time. What you're probably also getting at is the cells may also be, to some extent, seeing the mechanical properties underneath the supported lipid bilayer and whether that's basically having an impact. We've not done that here. In a completely different set of studies, we've been looking at how T cell basically activation, polarization is impacted by the viscoelastic properties of the substrate. And so that work is all with hydrogels instead of these mesoporous silica because we have a much better control. And there we do find that the behavior of the T cells is dramatically altered as we vary the properties from a viscoelasticity that would be more indicative of like a soft tissue to one that would be representative of a fibrotic tissue. So I think that's really important, but it's not directly incorporated into these particles.
Fabulous work, David. My question has to do with toxicity. Sodium azide is multiples more toxic than sodium cyanide, and if you dilute azide with a few atoms of carbon, it's still toxic. How do you keep the cells from the toxicity before clicking?
Okay, so these are basically chemically modified sugars. We do not see any. I mean, I imagine if we went high enough, you could eventually get some toxicity issues because pretty much anything will be toxic at a high enough concentration. We actually we don't see issues with toxicity either in cell culture or in vivo with this. And we've actually used this system quite a bit directly with these groups in vivo as well, where we're actually do the metabolic labeling in vivos versus not. So over the dose ranges that we're using for the type of labeling we're trying to do, we actually have not seen any issues. But of course, before this would move on to anything in humans, we'd have to do much more formal tox than we have done. But it does not seem to be a significant issue, and that's supported by a lot of other work with other investigators using these same kinds of sugar molecules. So it doesn't seem to be a big problem of the ranges that we use.
Hi, here. I have another question, so thank you for this great talk. My question comes into, so you've shown many or MHC class 1 on your artificial APC. Have you looked at any MHC class 2 or HLA class 2? And also, have you looked at the great polymorphism of HLA, so not only looking at A2, but maybe some BEC alleles or DR, DQ, DPs?
Yeah, and we've done, in terms of the antigen specific, we've done very little at this point in time. We've mainly focused on HLA2 just because that's the most widely distributed. And so I'm trying to think, we've done a little bit. We actually have an ongoing project with Kathy where we are taking some of the, she's been running clinical trials of neoantigen vaccines. Bob Langer mentioned that as well. Those therapies, when they're used at later stages of cancer, have not been hugely effective at this point in time, even though one can generate immune responses. So we've been doing some work with Kathy, seeing if we can use the system to actually isolate basically T cells from the patients and then dramatically expand them so that we can reinfuse basically many more from the argument that maybe it's just a numbers game, that a vaccine can't generate enough numbers in advanced cancer. So there we've played around a little bit, but not extensively. So we have gone beyond HLA2, but not in any depth yet. Yep.
Thank you so much for great presentation. I have a question about pro-inflammatory cytokine that you loaded on the nanoparticles.
Yep.
IL-12, IL-15s, they boost the immune systems. Have you ever see some sort of the autoimmune or extra activation of the immune systems, which is one of the problem of the right now cancer vaccines?
Yeah. So in terms of the first part of the question, this is bad. I do not remember the dose, but the dose we're delivering on the cells is actually quite small. So we've not seen any issues related to toxicity of the cytokines. And I just want to clarify as well, the cytokines aren't on nanoparticles. We basically use sugar nanoparticles to metabolically label the cells, and then we basically click on the cytokine afterwards to the cell. So we've not seen cytotoxicity with the doses we're giving. However, we have noticed these are relatively small doses and they're localized to the T cells. When we take the same dose that's on the T cell and we give it systemically with the T cells as a control, we do see some detrimental effects. So the same dose delivered a different manner is going to have pretty distinct outcomes. So we think that the advantages here will be, one, greatly reduced doses, and two, really controlling where the cytokine is being able to impact cells.